So the next portion is that we are going to be analyzing results. And so when we're looking at our results, there's different terms that I feel like come up a lot on the psych soc and biochem section, which is precision, which is how consistent the results are. So, and then accuracy, how close to the accepted value the results are. And so I think a common diagram that many professors use to describe this is this target example. And so precision, when how consistent the results are, would be like how many times you're getting that same result. And so C would be the best diagram to demonstrate that. And then accuracy, how close to accepted value. When you're doing a dart game, you're trying to get in that center. And so, B would be both a mixture of accuracy, so getting that into the center of the dartboard, and then also precision, because a lot of these darts are in that similar position and are pretty consistent. And then after that is looking at reliability and validity, which are more associated with the tool that you are using to analyze your results. And so reliability is the consistency of results from repeated experiments and the degree to which an assessment tool produces stable and consistent results. And then validity, the extent to which a measure reflects the phenomenon being studied, would be when that measure is actually showing the correct value it should. So there can be a tool that might be reliable, so it will give you the same consistent answer, but it might not actually be valid, as in it might not actually be giving you the correct result. But to be valid is when it actually reflects the process or the topic that you are studying for your experiment. And then you are also commonly looking at the significance from p-values when you are analyzing results and seeing p-values from t-tests and different statistical tests to determine how significant the differences are from your results of the experiment. Now we are going into the conclusion. So now is when we're reviewing our results. This is when we are comparing it with other past studies that have done like a similar type of experiment or anything like that. But the two main terms to look out for is reproducibility and replicability. So these are really important, I guess, for when you're doing experiments. So reproducibility is being able to obtain consistent results using the same data and code as the original study. So as in, if you were to do the experiment with your team, and repeat it, I don't know, 3, 4, 5 times, you would be getting the same results those 3, 4, 5 times. So just and following all of the exact same processes that you did the first time. On the other hand, replicability is when you're obtaining consistent results across studies aimed at answering the same scientific question. So let's say your lab gets a certain result, and another lab who's also researching the same topic decides to do the same experiment as you. If they get that same exact result that you did, then that would indicate replicability. However, sometimes something may be reproducible in one lab, but not necessarily have the same results in other labs, in which case then it wouldn't be considered replicable because the other research team is not getting the same consistent results as your team.